Hi, my name is Jonathan, and I'll be giving a quick overview of Arvados running on Curoverse Cloud. Arvados is an open source platform for managing, processing, and sharing genomic and other biomedical data. For this demo, I want to experiment with the GATK3 Haplotype Caller on a dataset of 10 whole exome FASTQ files to generate different VCFs. My goal is to run the same pipeline several times with different variant call thresholds. I'll use the outputs for more analysis later. To get this work done fast, I have to address a bunch of challenges with Arvados, and you'll see how I do that. I'll be working in the browser-based visual interface for Arvados, which is called Workbench. But you should know that everything I do here, I can do on the command line or with the API. This is the basic dashboard in Workbench, which gives me a quick summary of what is happening in my account. My first challenge is quickly organizing everything for this effort in one place, so I can easily keep track of it without having to scribble lots of reminders in my lab notebook. In Arvados, we do this by creating a project. I'm going to give my project a logical name, and then paste some notes, which summarize what I'm doing in this project into the description tab. This will be especially helpful when I share this project with colleagues. The next challenge is putting together the dataset I want to process. Before I had Arvados, I worked like most bioinformaticians and stored my files in a traditional file system using folders to organize everything. The problem is that every time I wanted to make a new dataset, I'd either need to keep track of the file paths or make copies. Generally, it was a hassle. Sometimes I lost track of the data, and I always wasted time doing data janitor work. In Arvados, we don't copy files. They are reliably stored in the cluster once. Instead, we make collections. A collection is like a virtual folder or a branch in a source code control system like Git. Collections have a lot of advantages over traditional folders, the first of which is that they are very fast to organize lots of big files. To get rolling, I'm going to make a virtual copy of two collections I already have from other projects. You can see as I do this that it's easy to find those collections based on their metadata, and copying them into my project is instant. We'll grab 20 exomes from an old unified genotyper project and 30 exomes from an old haplotype caller project. Next, I'll combine these two collections, and we can see I have a new collection now with 50 exomes and their related files, which take up about a terabyte of data. That operation happens just as quickly with 200 terabytes of data as does with just one. The final quick manipulation I'll do is to search through this combined collection to find the 10 exomes I want to use for this project and to generate a new collection. Now we're looking at the page for the collection I'm going to use for this analysis. I want to pause and point out a few features of collections. First, at the top, we can put some metadata about this collection. I'm going to give it a logical name. Then you'll see that in addition to this human readable name, each collection has a universally unique ID, or UUID, and a content address. The UUID is a consistent reference to this collection. It never changes. The content address is based on a hash of the data in the collection, so when I use it to retrieve the data in the collection, it automatically verifies that all of the bits are still there exactly as they were the last time I used the collection. If I make any change to the collection, such as adding or removing a file, Arvados will keep track of each version, and each version will have its own content address. At any time, I can retrieve any past version of a collection with the content address, and I can ask any collection to tell me all its versions. Arvados is designed to make all of these operations extremely reliable and durable. Of course, if you're like me, you may use tools that want to see files as if they're stored in a regular file system, and Arvados can do that too. I'll show you a cool trick. I can copy the content address or UUID for this collection, go over to the command line, and just mount it as a drive. Now I can read data from the collection as if it were a normal drive. Because it's virtual, I can define any directory path structure I want. Here, I'll just do a simple ls on the collection, and you can see the 10 exomes. Okay, we've gotten our project organized and solved our data management challenge, so let's move on to the next problem, actually running a pipeline a few times to try out different configurations. From back in the project, I can just hit the button to run a pipeline. Again, I can easily do this on the command line as well. In Arvados, you define a pipeline by defining a pipeline template. I'm not going to do that in this demo, but it's easy to do, and you can use all the tools and scripts you normally work with. Instead of making a new pipeline template, I'm going to run a pipeline I already created. I'll just grab it from a project that we use in my lab to store all the pipeline templates we like to share. Before we had this, sharing pipelines, especially new pipelines, was a big hassle. Each time you run a pipeline, Arvados records that run as a unique instance of the pipeline, and you can set the input parameters. For this pipeline run, I'm going to give the run a logical name. Then I'm going to pick the input collection we made. Finally, I'll set the variant threshold for this run. I'm going to leave all the rest of the configuration settings at their defaults. Just so you can see it, let's take a quick look at a summary of all the components in this pipeline. You can see in this list that this pipeline runs 9 jobs. Each one uses standard tools. You can see for each job I have a content address for the script for that job. Arvados stores a copy of all the scripts you run in a Git repository, so it can reliably rerun any pipeline, and these addresses are from that Git repo. I should also note that for some of these jobs, I created Docker containers. Docker containers are a very convenient and easy way to make sure that when a script runs, it has all the runtime libraries installed properly. All right, let's run this. When I click Run, behind the scenes, Arvados is doing a lot of work for me. The pipeline processing engine is scheduling compute nodes for each job. It's installing the Docker containers and scripts, hooking them to storage, tracking the processing, and recording everything that is happening. If I need the details, I can pop over to the Logging tab and see the real-time logging feed. We'll use some demo magic to jump ahead. You can see here that this pipeline took 1 day and almost 7 hours to run. In CPU usage, though, it took 7 days and almost 5 hours to run. We could do this because some of the jobs in this pipeline do analysis that can be easily broken down into smaller tasks and run in parallel. Arvados handles this kind of embarrassingly parallel computation easily. My last challenge is to run this pipeline again. That's easy with Arvados. I just hit Rerun. I've got some options if I want to run the exact same pipeline again, update the components, or run the same pipeline with new components. I'll run the same one with different inputs. New name to keep track of this instance. Same input files. Different variant call threshold. And run. For this second run, Arvados does some magic itself and just skips the jobs that have already run before and don't have any changes or use random numbers. We'll use some of our own demo magic again to skip ahead. In the past, one of my biggest challenges was figuring out where data I had came from and just seeing the history of my pipeline runs. Arvados records this kind of information automatically. I can go to the provenance or usage graph on any pipeline or collection and see the history and usage. Here it is for this pipeline run. The rectangles are data inputs and the ovals are jobs. If I scroll down to the bottom, I can see the output collection. One click and it puts me into that collection with the VCFs I generated from this run, and everything is already conveniently named. With automatic provenance, I can do another thing that is very helpful, which is to compare pipeline runs. If I go back to my project, you'll see I now have a record of the 2 pipeline runs conveniently saved for me. I'll choose these 2, hit compare, and then I can see in the provenance graph the jobs and data that were identical and where the 2 pipelines diverged. Okay, we've solved 3 of our big problems: getting our work organized, managing and tracking our datasets, and running and rerunning pipelines. We've got a last challenge for this project. I want to share it. Let's go over to the sharing tab for this project. I actually want to share the project in 2 ways. First, I want to give my colleague Jane write access so she can help me extend this work and do some other runs. Second, I want to give read access to anyone on the internet. Normally, I would only do this when I wanted a final URL to publish in a paper or for reviewers. But for this project, I want to show it off right away to a larger community of people, some of whom don't have Curoverse Cloud accounts. I'll go set up the anonymous user group and give it read access. Now I have this URL I can send to anyone. If I go to a browser where I'm not logged in and paste in the URL, you see that anyone can access the project with just the URL and no login, so they can download data and look at how I did the work. It's also possible for someone to copy the whole project into their own Curoverse Cloud account or even to another cluster running Arvados, for example, one in their own data center. Of course, Arvados copies and cryptographically verifies everything, so when they go to run my pipeline on their cluster, it will run just like it did in my account. All right, we're done. Not including the processing time, That took less than 10 minutes of work. As you saw, I was able to tackle all of the challenges I faced doing this haplotype caller work quickly and easily using Arvados. Our goal is to streamline your work and make you more productive. Hopefully, this got you interested in trying Arvados. Sign up for a Curoverse Cloud account and give it a run yourself. You'll find a bunch of public data, pipelines, and tutorials to get you started. I look forward to hearing what you think.